Hi everyone, I'm Bruce. I want to take this opportunity to talk about some of the science behind my recent research at the University of Auckland, which has been generously funded by the Neurological Foundation. So we've developed this ultra-thin plastic bioelectronic implant, which we can position along the spinal cord over a spinal cord injury to be able to deliver a healing electrical field treatment with electrodes. So we can also use electrodes to record electrical activity from the spinal cord. And we hope to be able to do this to explore whether we can discover biomarkers for injury and subsequent recovery after a treatment. Just a quick slide to show a bit of spinal cord anatomy. I realize that a lot of the talks will be about the brain this week. So the spinal cord sends signals to and from the brain and sends signals to and from the brain to muscles and to the rest of the body. So looking on the right, depending on where an injury occurs along the spinal cord, you can have loss of different types of function. And the other thing I just wanted to mention is the brain has a dura mater membrane, which forms a blood-brain barrier to stop anything from reaching the brain that's not supposed to. And this extends down the spinal cord as the dura mater, which contains cerebrospinal fluid in which the spinal cord is suspended. This slide just shows some stats about spinal cord injury in New Zealand. So we have, like other Western countries, we have a high incidence and prevalence of spinal cord injury. There's a recent publication in 2022 that looked at the data between 2007 and 2016. They found that the major causes of spinal cord injury in New Zealand are from car crashes and falls, but also sports is sort of the third most common occurrence. There were 217 cases of new spinal cord injury in 2018, and this research showed that there's a general about 6% increase in these numbers per year. So also quite interestingly, the demographic of spinal cord injury is changing. So 10 or maybe 20 years ago, the typical spinal cord injury was thought to be in sort of young males, but this is really changing with the population getting older and the disparity between the genders changing as well. Spinal cord injury is a complex injury to treat. Another thing that's important to note is, as researchers, we quite often really get hung up about trying to restore motor function, whereas spinal cord injury can result in neuropathic pain as well as loss of bladder and sexual function. And these can often be just as difficult impacts on quality of life as the loss of function, especially in the older patients. Spinal cord injury usually, or a traumatic spinal cord injury normally starts with a primary injury incident, which causes a wound on the spinal cord, resulting in cell death. And this causes various cascades of things to happen over the following hours, days, and weeks. So for instance, the tissue around the injury begins to have issues due to the loss of connectivity through the spinal cord, and there's sort of a flooding of neuroinflammatory cells such as microglia and astrocytes, and often a lot of cell death and loss in these areas, which sort of spreads the injury. After several weeks, there's usually formation of a glial scar around the injury, and which sort of prevents further spreading. And one of the issues with this is when you then come to treat a spinal cord injury and try to get regeneration to happen around there, this glial scar often prevents growth back through the injury site. So the type of treatment that we want to try and deploy from our implant is an electric field treatment. This is where we place electrodes either side of the spinal cord injury. and use them to generate an electric field that penetrates through the tissue. So electric fields can actually encourage axons to grow, but it could also act as a navigation system. It tells the axons and nerve fibers which direction to grow, which is we want them to grow across the injury. I'll just mention quickly that we use a quite unusual type of electrical stimulation compared to other applications that are in use. So this is a very slow, low frequency electric stimulation that's a lot more like direct current. So the bottom left figure shows the idea of what we're trying to do with electrodes either side and to achieve an electric field. In the red box, which is blown up on the right, shows the tip of a severed axon. So when these axon tips come in contact with an electric field, a growth cone forms, so this sort of enlarged bulb, and the growth cone helps the axon to grow towards the negative cathode electrode of the electric field. I just want to mention really quickly that there are some other kinds of electrical stimulation applications that are already in the clinic. On the left is neuromodulation. So this is where a stimulator is placed just past an injury site and stimulates the spinal cord past the injury to create the kind of activations that happen with a walking gait. So this kind of device can sort of artificially create a walking gait and can actually restore walking function to people. So if you've not heard of this, you should look it up. It's a really incredible technology that's been recently developed and deployed. Also, spinal stimulation is used to control neuropathic pain, so to inhibit pain signals, chronic pain signals that people get after spinal cord injury. And then on the far right, there's also some applications now of deep brain stimulation, where you insert electrodes into the brain and stimulate areas to try and treat symptoms of conditions such as epilepsy or the tremors that you get from Parkinson's disease. So the point I really want to make here is that all of these other devices use a very different kind of electrical stimulation. It's very fast, high frequency pulses. So sometimes usually hundreds or thousands of pulses per second between the electrodes. And the reason that this is done is that kind of stimulation can cause neurons and axons to fire. So it activates the part of the brain or the spinal cord in which the electrode is placed. The type of stimulation that we're using for the application of wound healing is completely different. There's the change in pulse polarity only happens over minutes or sometimes even longer. So it really is just a direct current stimulation. And actually, these other devices cannot even produce a stimulation like this. They're hardwired to produce only very rapid bursts. And I'll tell you a little bit more why in a bit. This low frequency stimulation that we use for wound healing has been successfully used before to treat spinal cord injury in the 80s to 2000s by a very prominent American researcher called Richard Borgens. So in the late 70s and 80s, his group showed that in a petri dish, you can apply an electric field and it will cause axons to grow, but also you can control the direction of growth using the electric field. His group over the 80s then installed electrodes first in fish and then in guinea pigs with spinal cord injuries, and showed that there was regeneration of tissue around the injury, but also through the injury site. And several other independent groups did a similar experiment installing electrodes in rats and got similar results. In the 90s, the Borgens group started a preclinical trial in which they installed stimulators and electrodes in patient dogs. So these were people's pet dogs that had recently had a spinal cord injury and were able to be enrolled for the study. After 6 months, they were able to show that with the stimulation, there was noticeable improvements in pain sensitivity of the animals, and even some small improvements in motor function in some of them. This led to stimulators and electrodes being installed in clinical patients in 2005 and 2014. And these studies achieved pretty similar results. They showed improvements in fine touch and pain sensitivity, as well as improvements in motor skills compared with baseline patient data. So some of these improvements were pretty modest, but even very small improvements are really great, incredible achievements when you're treating spinal cord injury. Any improvement in the quality of life can make a huge difference. However, there's some problems both with the applications and the technologies that were used in these studies, and I'll go into that for a little bit in the next few slides. So firstly, the electrode position that they used was generally electrodes were placed very far away from the injury site. So in the bottom left figure shows in these studies the position of the electrodes represented by the colored circles, and the large circle in the middle that's red represents the spinal cord injury, and the red rectangles nearby indicate the position that the electrodes will be in our study. So the reason that electrodes in these previous studies were placed quite distant from the injury was they were worried about doing damage to the spinal cord. So a lot of current was being put through these electrodes, and a lot of heat, and actually in several instances in these studies, it was shown that there was some damage to the muscle tissue near the electrodes, some burn damage, that sort of thing. With our application, we're using very small electrodes that are directly on the surface of the spinal cord. And due to their position, we can actually put a very low current through these electrodes, an order of magnitude lower than was used in the previous studies, and we're still able to achieve a much stronger electric field. So there's no risk at all of overheating or causing any damage. This sort of concept is shown in the figure on the right, which is from a computational modeling study from 2014 that compared the electric field produced on the surface of the spinal cord from electrodes on the surface on the right column and that were positioned above the dura mater membrane in the left column. And we can see on the left column that the electric field, the red heat map and blue heat map shapes shown on the surface of the spinal cord are quite diminished and spread out. So when the electric field has to travel through these layers of membrane and cerebrospinal fluid, it diminishes the field, it spreads it out. And more importantly, on the bottom row, we can see a section that shows inside the spinal cord. And the further away position of the electrodes also results in very little penetration into the spinal cord, whereas on the bottom right panel, with the spinal surface electrode, we can see that the heat map goes a small distance into the spinal cord. And this is really important for the application that we're using here for wound healing, as we want that electric field to penetrate into the spinal cord around the spinal lesion. Another separate issue is to do with the degradation of electrodes. So the lower frequency electric field that I was talking about on the previous slide that we use for the wound healing results in a Faradaic reactions in which ions are exchanged between the tissue and the electrode. So ions in the tissue are used to recharge the electrode, whereas metallic ions from the electrode are passed into the tissue, and these are sort of toxic byproducts that are released into surrounding tissue. So this was something that wasn't really addressed or checked in these previous studies. In our application, we're very lucky we have access to this cutting-edge electrode material that's been recently produced by our collaborators in Germany, who also design and fabricate our bioelectronic implant. So it's a sputtered iridium oxide layer that we can place over our electrodes, which slows down this exchange of ions and makes them much safer to use. So how it works is we can alternate the stimulation that we use between several different of these electrodes, which gives each electrode a rest period, which allows them to recharge and prevents them from releasing any toxic products. We've also used some computer simulations to decide how much current that we use in our electrodes and how to develop the size of the electrodes themselves. So this is a computer simulation of the spinal cord as well as the implant and the electrode position. And we're able to calculate how much current we should put through the electrodes in order to achieve the electric field that we want, that's strong enough to have the sort of healing beneficial effects in the tissue. And on the top right is our PhD student from Germany that is helping us with this project, Lucas Matter. So this is our implant, what it looks like. In the middle panel is the entire implant, and that whole thing is only a few centimeters long. On the top is a blown up version of the end of it. So this is the part of the implant that contacts the spinal cord. You can see tiny black dots along the outside, which are little recording electrodes. Each of these is only 60 micrometers wide. So you basically almost can't see these with the naked eye. And then we have some larger blue electrodes, which are specialized for doing stimulation. The implant is incredibly thin. It's made of a very thin polyamide plastic, only 8 millimeters thick. And this allows it to conform to the surface of the spinal cord, and it doesn't take too much space in that subdural area where the cerebral spinal fluid is. And there's a couple of diagrams here to show what this thing looks like when it's deployed on an animal, a rat, and the size of the thing. So moving on to some of the actual work that I've done with this implant in the last couple of years. So the first thing that we wanted to do was I developed some surgical methods to allow me to install this implant along the spinal cord underneath that membrane. And then we tested this out. We used two different designs of the implant. So the bifurcated version has two arms at the end, which sit either side of the spinal cord, the hemisphere. And we also had a planar version, which was just all one piece. And then we had a control group that just had the spinal cord exposed, but no implant. And we then installed these in rats, and then we checked their hind leg function over a week, and then looked at the spinal cords to see what they looked like after a week of this implant. And what we found with the bottom left graph is some of these animals had a very mild but transient motor deficit, but were returned to normal function after 7 days. We also kept a couple of animals for longer, for 5 weeks, but didn't see any deterioration of their ability to move their back legs, to walk normally. Looking at the spinal cord tissue on the sort of top panel, we found that the spinal cord was the normal shape and size compared to controls. And we also looked at some markers for foreign body response to see if there was a migration of astrocytes and microglia towards the surface where the implant was. And we saw no real differences between the at least the bifurcated group and the control group. So there were some small differences in the foreign body response for the planar implant. And so we proceeded after these experiments just using the bifurcated version, as it seems to be a bit more biocompatible. After this, I developed a external housing to allow us to have a plug on the far side of the implant, which would enable us to plug the implant in and access those electrodes for stimulation and recording. So this is shown in the middle panel. It's made of 3D printed plastic. And this allowed us to install these implants in animals for a bit longer. So we had them in for up to 3 months, and then checked the function of the animals, and we had no health issues or motor issues in these animals in that time with the implant installed. So they're completely normal and healthy animals. Importantly, we also wanted to check the condition of the implant after it's been inside tissue for 3 months. So what we found was that the implant was still in the correct position along the spinal cord, which is shown by the CT scan image on the bottom left. And then we also had a look at the polyamide plastic electrodes and electrode tracks on the implants after they'd been in the tissue for that long. And we found them all to be in pristine condition, so there was no sort of degradation of the implant. So this is really important for us because we want to test these regenerative treatments using an electric field. So we need a long enough period to be able to apply those treatments, and then a long enough window to be able to see if there's any effect on the injury, to see if there's any functional improvements. And so a period of 3 months in which we can install the implant is perfect for us to be able to do this. So this experiment also allowed us to do some recordings of spinal cord activity, and these were actually a world first recording in freely moving unrestrained animals. So this video shows a 6-second segment recorded from the animal in which it does 3 different behaviors. So first of all, it walks or runs for a couple of seconds, and then it sits still, and then it rears up and it stands leaning up against the edge of the environment. And what we found here was that this produces really interesting electrical recordings along the spinal cord, which we really don't have too much to compare with. So we're still investigating this, but I just wanted to show you a little bit of what this data looks like. So on the right, those 6 traces are from 3 sets of 3 electrodes recorded from either arm of the implant. The black trace is filtered to show only low frequency activity. So normally when you record brain activity, this kind of filtered activity shows network activity that's happening around the electrode. So many neurons firing, for instance, like gamma or alpha waves that are looked at in sleep. The red traces show high frequency filtered activity, which is in the window for us to be able to see spikes recorded, action potentials recorded from single neurons or axons or compound action potentials from a small bundle of axons. And what we can see is when the animal's running, the low frequency activity, the black trace sort of oscillates up and down like mountain peaks. So this most likely corresponds with the hind leg gait of the animal. Because we have our implant placed on the thoracic spinal cord, we're only recording activity from the back part, from the legs. At the same time, the high frequency spikes, they seem to be a little bit more denser in the peaks of these oscillations. And this is something that you often see in brain activity. The firing of the single neurons corresponds to the network oscillations of the low frequency part of the signal. When the animal's sitting, both of these traces are quite muted. But interestingly, when the animal is standing up, we get a huge influx of the red high frequency spikes. So this was a little bit of a surprise to us when we first saw it, but I think it's because the animal is putting all of its weight on the hind legs, and so there's a lot of sensory signals coming from the area where all of the weight of the animal is on. So we haven't really been able to dig as far into these recordings as we might like. So we have a PhD student, Brittany, who has started investigating for us. So the first thing that she wanted to look at is this figure on this slide shows the high frequency activity over 5 seconds for all of the electrodes on one arm of the implant. And the first thing you notice is that the signal looks really similar. It looks like the same signal on every electrode. So if this is in fact true, if they're all occurring at exactly the same time, this would mean that they're probably noise caused by muscle activity or movement. So Brittany had an investigation and look at these, and what she found was that on each electrode, these signals are slightly offset from each other in time. And the reason is because this is the same signal that's being recorded at propagating down the spinal cord. Because we have rows of electrodes over about 1 centimeter. The difference in the signal between electrodes shows the time that it took to reach that electrode, if that makes sense. So on the top right is shown a lot of examples of this. On the bottom left, So once you detect the time position of each of these individual axon spikes on each electrode, you can draw a line to calculate the gradient, and this is the velocity that the action potential is moving. And so on the top right, Brittany's calculated the velocity for a bunch of examples of different spikes. And on the bottom right is the population of a large number of these spikes, the velocities graphed for sort of a population over a whole recording session. And this experimental data is shown in blue, whereas the orange data is a control data set which has been shuffled so that the position of each signal has just been randomized, so that we can see what the population would look like if the position of the electrode didn't matter. So just looking at the blue diagram, which is sort of towards the left, the majority of these signals had a velocity in an afferent direction. So this is signals that are going from the body to the brain. Whereas on the very right-hand side of the graph, we have velocities that would be efferent signals, which would be from the brain to the body, so would be motor signals. So this proportion of the sensory and motor signals, it fits the area of the spinal cord that we're recording from. So because we're recording from the dorsal surface, the majority of regions near our electrodes are sensory, but there are also some deeper motor areas that are allowing us to record some signals that are going in the other direction. Another thing that we're trying to develop in our School of Pharmacy group is the ability to deliver drug treatments from the implant. So because our implant is positioned on the surface of the spinal cord below that blood barrier, brain barrier membrane, it gives us the ability to use it as a platform to deliver medicines. So we're developing ways that we can install drug molecules along the implant and then have them be slowly released. So PhD students Ernest and Svenja are working on these kinds of technologies. So for instance, Svenja's been working on a hydrogel, which is a jelly-like liquid in which we can inject drug molecules inside. And then when it's injected along, say, into the spinal cord environment with a different temperature, this hydrogel will change state and slowly release drug molecules over days, up to sort of 7 days. And you can even modify the delivery period longer or shorter by changing the elements that are used to construct the hydrogel. Another type of technology we're looking at is conducting polymer. So this is more of a kind of metallic structure that we could place on the implant, and we're able to incorporate a hydrogel inside of it. And then when you would electrically stimulate the conducting polymer, you would be able to control the release of portions of hydrogel, which themselves can slow release drugs over a period of a week. So using a combination of these kinds of technologies, we may be able to release drugs for an even longer period than a week. And so the idea is to develop these things and then find a way to put them onto the implant, and then we would be able to try combination treatments of electrical field as well as drugs such as neurotrophins, which have been shown to be effective at enhancing regeneration after spinal cord injury. This is really a summary slide that I wanted to talk about the process of this research and where I think it's going at the moment, from conception of the idea towards the ultimate goal of this is we want to develop a clinical device that can be used to help New Zealanders and their whānau with spinal cord injury. So at the very top, designing and fabricating, testing an implant. So this was done before I joined the team, and the green text indicates the objectives that I've been working on during this Neurological Foundation project. So really it's been about developing surgical methods, testing whether the implant is biocompatible, whether it could be installed okay without causing any issues. And then we moved on to developing this external connector, which allows us to make recordings and will allow us to provide electrical stimulation. We also have combined it with a spinal cord injury model to make sure that the implant can still function in that environment, and we're able to install it for up to 3 months, and we're able to make these recordings. So really the next step here is we're perfectly poised now to test low frequency electrical field treatment, and that's actually work that I'm lined up to do hopefully in the next few weeks to get that started. We also want to try a few different types of electrical field treatment, and then at the same time we're really working on developing capacity to deliver deliver medicines from the implant. Sorry, the transcription thing's got me a little bit distracted there. Another thing we want to do in the future is to develop a fully implantable system, a little bit like they used in some of the earlier studies. So at the moment we connect to the implant to deliver electrical stimulation, but if we can develop an implantable stimulator, we can install that in a subcutaneous pocket, and it can allow sort of much longer periods of stimulation. And we also want to test the combination of the neurotrophin medicines and electric field treatment. Past these sorts of points, it's hard to plan exactly how we want to develop this because things can change. But my current thinking is that we want to upscale this implant to be able to be the right sort of parameters to be installed in clinical patients. So we may use like a large animal model for this, sheep or pig or something like that, just to try surgical techniques, because the human spinal cord and the dura is quite different, and it's important to really get these things right to find a really watertight way of installing this implant with the advice of neurosurgeons, which we're currently sort of in initial talks with. And then really we want to move forwards to being able to test this in a clinical trial. And what I would hope is that that kind of thing is maybe 6 to 8 years away. Just to give you an idea on what we think the timeline might be for developing this sort of device, which is really at a preclinical testing level at the moment, to something that we might be able to use in people. So thank you very much for listening to that. I just want to acknowledge all of the collaborators that I have working on this project, and also the funders, obviously Neurological Foundation and others. It's not only me working on this project, but we have quite a large interdisciplinary team. So we're very lucky we have pharmacists. I myself am a neuroscientist or from a psychology background, but we also have input from engineers, coders, and all of these people make this kind of quite large vision project possible. But thanks again. And I'll be happy to answer some questions, of course.
Yeah, thank you so much, Bruce. That was fascinating, and it was really amazing to see the journey that the implant has taken. Like you said, there'd been the studies earlier in the 80s and 90s, and how that's informed what you're working on now. So it just seems like it would be such an amazing thing for patients in whānau with these injuries. So thank you for your amazing work. As Bruce said, the Q&A is open now. So we've got our first question here. Please feel free. Oh, also I have to apologize. I forgot to share my slides at the start when I did my housekeeping. So apologies for everyone who was looking for what I was talking about. Our first question has come through, and it is, how do you pull together the research group with so many different disciplines?
It's a good question. I think, to be honest, it's really taken a bit of time to do that. So my my friend Darren Sviskus leads this project, and he's the one that first recruited me at University of Auckland. Really it's just been through getting in touch with people at different conferences. I think that's how we originally made this connection with the University of Freiburg that fabricate these and create these implants with us. And we're very, we started doing these spinal cord recordings, and we really struggled to understand what we're looking at. We didn't have the expertise within our group at the time to really understand that. And so we've looked to recruit people from the Auckland Bioengineering Institute, and that's where we have a PhD, Brittany, who's got the background there in programming and understanding electrical signals. It really is. It really is a challenge to collect these different people and then in order to keep them as well.
Yeah, I'm sure. Yeah, wow. And I suppose going on down the track as well, there'd be connections in with physios and things like that when it becomes a clinical application. So it takes a village.
Yeah, I mean, that's a really good point. So one thing that we've been doing, especially in the last year or so, is trying to connect with the wider sort of spinal cord injury community. So we managed to host a symposium at the end of last year, that brought together spinal surgeons from Middlemore and from Christchurch, and also some rehabilitation people that have spinal cord injury to talk to us, as well as researchers, and just really show some of this research and get ideas from different people. And also talking to spinal surgeons is really something that we've tried to do in the last year or so, and we really want to follow forwards with that. So we are quite a long way from having a clinical device, but you really need to think about these things now and how the direction of your research is heading towards that, so that you don't sort of go the wrong way or hit road bumps because you haven't really thought about the translation.
Yeah, absolutely. Okay, next question is, is this device or this implant, has there been any research into, or will this be effective for old injuries?
Oh, it's a really good question. The majority of patients have had an injury for a while. And these are the people that ultimately we want to help. Probably us, like most other researchers, we start trying to deploy this thing in recent injuries, and it really is it's partly because it's the most likely way for it to show effectiveness, to show larger effects, and it's just because when you treat a complex injury like that, like this, you can sometimes reduce the effect of those later cascades that happen after days and weeks. But ultimately, we want to, if we can find a strong effect of this electric field treatment, we want to test it in chronic older injuries. And I've sort of had a little bit of a look at the background literature to do with that, and there's no reason to think that it can't be effective in older injuries. So there's some recent studies that show in older spinal cord injuries, there is quite a lot of sparing of axons. So there's whole regions near the injury that are inactive, but are not actually functionally damaged. So it may not be too difficult to bring back function to those regions. Of course, there are other areas within the injury where we're looking to try and regrow new fibers across the injury. And we really hope that we'll be able to do that with this sort of electric field, but it is more of a challenge. But yeah, I mean, that would be the aim, is to definitely look at that.
Yeah, absolutely. And I think this question, I'm not sure if you'll be able to answer this question, but I'll ask it anyway, do you think this might have applications for people with MS who have multiple lesions on the spinal cord? Now, I appreciate you're not an MS researcher, but...
No, I think we're starting off with a very simple model where there's just one injury site, but the reality of spinal cord injury is there are often multiple sites. Especially, for instance, in a car crash or a fall, it's often several places where there's damage. So there's no reason to think why we can't have a sort of a longer implant that applies to multiple places. So we have a very generic implant that we want to use at the moment, and we have an injury that we apply. It is quite a good representative impact kind of injury, of similar to what most of the human clinical cases are, but we very much control where it is, and we place our generic shaped implant around it. What we want to be able to do in the future is to produce a custom version of this implant for each individual. And this is something that would be completely possible to do even at the moment, if we can show that this technology works. So there is technology at the moment that would allow us to quickly redesign different versions of this implant and then produce them. And actually our collaborators in Germany are soon to get hold of this sort of technology. And, you know, for ourselves in the short term, we're very excited about being able to make changes to the implant very quickly and get new versions to test different things. But I think this sort of approach is the sort of thing that could be used to customize and personalize this kind of treatment to different individuals. And for instance, you could have electrodes positioned to address multiple sites on different parts of the spinal cord.
Amazing. It's remarkable. Your own personalized, bespoke implant. Okay, how difficult is it to find funding for this kind of work?
I mean, I think it can be difficult. The great thing about the funding that I've received so far about from the Neurological Foundation is to let me sort of develop this idea, well, from basically just an idea. We had an implant, but you know, what I've been able to do is basically show that it can be effectively used as an implant. And this makes the whole project much more exciting and attractive in terms of getting more funding. In terms of spinal cord injury, there's the Catwalk Trust in New Zealand that generates funding specifically for spinal cord injury. So they've also been very helpful to us in terms of supplying us with some funding. But there's other agencies such as Health Research Council and a few others as well. I think it can be challenging, but this is an area that really is important to a lot of people. And I think we're also able to show that the research that we're doing is really a sort of a cutting edge, you know, in terms of what other places in the world are doing as well. So.
Yeah, absolutely. And again, it's that collective effort. Like within your research, you've got all the different sciences in the group, and then you've got the collective funding efforts. It's really amazing. Just going on from that, is there any specific advantages to doing this in Aotearoa, New Zealand? I know you're collaborating with the guys in Germany. So is there any particular reason why it works well here?
Oh, that's a really good question. I think I would say that New Zealand really does punch above its weight in terms of world research. This is a great place to do research. We have a good attitude. I think, you know, it's a little bit cheesy, but, you know, it's sort of in our nature to just kind of get things done and to, you know, build speed record breaking cars out of duct tape, that kind of thing. And, you know, in the time that I spent overseas, I really saw, you know, the value of that kind of attitude, of that we can quite often get things done without a huge amount of money and resources just by, you know, being imaginative and thinking outside the box. But I think this is a good place to do research. It's my home. I'm very happy to be back here. And I think another thing is, in the modern age, it's really easy for us, for instance, to collaborate with people in Germany and America. And so, you know, we're in a good position to be able to still do these international collaborations, even though we're quite a travel distance away.
Yeah, absolutely. It's nice to hear that you're glad to be back in New Zealand doing your work, and we are very glad to have you in this remarkable project going on. following on from that, another kind of career question for you: has there been a particular highlight for you in your work to date?
I have to say I'm very partial to the spinal cord recording. So I have a background of brain recordings. This is really what I worked on overseas, was in stalling devices in the brain to record brain activity while the animals are freely moving and behaving. So we're really trying to understand how behavior relates to brain activity in different areas. When I came back to New Zealand, I sort of jumped across to an implant that can record spinal cord activity. And, you know, this is a really novel implant compared to what I was doing with the brain. There are many, many groups doing these brain recordings, and it's been done for the last 50 years. This is very, very much more exciting. And I think, you know, having a look at some of these brain recordings, these first, spinal cord recordings that we're able to make, I really get that feeling that all scientists want to have, where you realize that you're looking at something that nobody else has ever seen before.
Yeah.
And, you know, that's really exciting. And, you know, we're still trying to understand it completely, but yeah, it's really nice to be in that position. So there's been other groups that have done recordings in spinal cord, but always in sort of anesthetized animals or in conditions where it's very highly controlled. But to be able to just allow normal behavior and movement, but see what's happening in the spinal cord is pretty cool.
Yeah, amazing. This is quite an interesting question, are there future applications that you can think of for people who've had a stroke, dealing with the signaling between brain and arm or leg, and it's no longer working? I guess you kind of touched on this at the start with the other electrical impulses in the spinal cord being used. But
there's one thing that I'd like to respond to. I don't think it's directly addressing the question, I'm sorry, but one thing that I've sort of realized and learned thanks to Brittany's literature research lately so she's looking at these spinal cord recordings and thinking ahead, is there's huge application for these sorts of recordings in terms of brain-machine interface. So if you were to develop some sort of prosthesis or like cybernetic hand or something like that, and you need to be able to stimulate with an electronic device in order to control it, the spinal cord is the best target for that sort of therapy. And this is because when you want to move your hand, there's a lot of different areas in the brain that activate towards that happening, but they're all in disparate parts of your head or different parts of your brain. But all of those areas, the signals comes together in the base of the spinal cord and then propagates down. So if you were wanting to control signals for moving your hand in a certain way, the spinal cord would be the interface for that. And I think that's a little bit answering what you're saying in terms of you know, controlling loss of function of arms and something like that after stroke. Is I think over the next 30 or 40 years, we will see development of these brain machine interface devices that allow you to use your brain to control a device that controls some sort of function. And obviously there's some high profile things being tried by crazy people like Elon Musk to create some sort of brain thing, although, you know, from what I see, that's not necessarily going too well. But there's a lot of groups working on that kind of thing, and, you know, it's the future.
Yeah, absolutely. And you're right, there's something to be said for taking your time and really looking at the research of these kinds of things. So that's maybe 30, 40 years in the future, but what do you think about spinal cord injury treatment in 10 years' time? Considering you mentioned the implant itself may be in a clinical practice or trial, how do you see it?
I think that, you know, I really do think we're going to start seeing applications that are working, at least in terms of showing modest or even moderate improvements. So there are a lot of really promising treatments around the world, but they're at early stages. And, you know, I feel like our device, our application is one of these types of things. One of the advantages of working on this device is it could be used to deploy other kinds of treatments. And this is really why we have this interest in trying to develop ways that we can release medicines chemical drug molecules from the implant as well, is a lot of the treatments developed by other people have been shown to be sort of effective, even gene therapy using reprogrammed cells, that kind of thing. But it's hard to deploy these things. Often in an animal model, they're injected into the spinal cord, and it's very, you can only do this one time. But what we would really like to establish with a device like this is a way to have access to that spinal cord injury over a longer period, over 8 months, where you could deliver drugs on a daily basis if you had the right technology in order to do it. So in a way, that we're also trying to develop this implant as something that could be used to deploy other treatments as well. Although we do have these very promising treatments that we want to try ourselves first.
Yeah, absolutely. Okay, well, that is the end of our questions. We did just have one comment come through that was thanking you very much, Bruce, for your excellent work and your excellent answers to the questions. I think there was some really interesting things that came through there, and I mean, this is amazing and one to really watch for us in terms of the tangible impacts that it could have on patients. So you and your team just keep it up.
Thank you.
You're very welcome. And thanks everybody for joining. Another comment, thank you so much. So thanks everyone for joining. The recording of this will be up on our website and YouTube later in the week, and keep an eye out in your letterboxes and inboxes for our latest Headlines magazine, where you'll see a bit more about Bruce's work as well. So, ka kite everyone, and have a lovely night.